This is the cytoskeleton, microtubules, centrosomes, and MTOCs. In this video, we'll particularly pay attention to how microtubules are organized. Remember this analogy that each microtubule is a straw, and there's nothing that holds one straw to another. We need something else, some sort of compartment or other protein organizing location to hold them together. So the question is, how are microtubules organized? They're organized by a microtubule organizing center, or MTOC. In this figure, you can see a cell which is stained for microtubules, and as well as for DNA in red, microtubules in green. And so you can see that there's a lot of microtubules emanating from many different locations. You can see kind of a core overlap right here. So this might be one of the microtubule organizing centers. Basically, what the MTOCs do is they will control the location, orientation, and number of microtubules within a cell. There's three examples of different sorts of MTOCs. The most common that we'll hear about is the centrosome. And so the centrosome is found in non-dividing cells, and you can see the centrosome is the circular structure here. It's light green, and it has some black kind of rectangles within it. The light green structures, that's gamma tubulin. It's a matrix of proteins, and there's also these two black centrioles, which are located perpendicular to one another within each centrosome. And together, each centrosome is made up of gamma tubulin, which is the primary part of this protein matrix, and then two centrioles. In dividing cells, you can see that there's a pair of centrosomes, so those are on each side of the mitotic spindle, and each of these is a MTOC, specifically the pole of the mitotic spindle. Here and here, each of those are MTOCs. Then a third type of MTOC is the basal body of cilia or flagella. And if you look over here, we can see that we have microtubules along these ciliated portions of the cell, and then they're anchored by the black protein complex, and so those are centrioles. And basically, a basal body is just a pair of centrioles. There's no gamma tubulin. And basically, the basal body is just a fancy name for the pair of centrioles. Now, I want to point something out, which is that when we talked about intestinal epithelial cells, we also saw these ciliated structures called microvilli, and we highlighted that there was actin found within them. There can be both actin and microtubules within the ciliated structures, such as microvilli. However, they're going to be attached to different things. As you can see here, the microtubules are attached to the basal body. This is going to be one long microtubule emanating all the way across this entire portion of the cell. On the other hand, for the actin, then that is anchored to the cell cortex, and so there can be a whole meshwork of actin. And so if we were going to have actin here, then that would be something like right along here, just on the inside of the cell membrane. This is figure 17-12, and here we're looking at the centrosome structure, which is also titled here as the anatomy of the centrosome. So basically here we see the whole centrosome closer up. So it's that green circular structure, which has a pair of centrioles, which are here colored in light blue. And what we're seeing that is an extra piece of information that's very important is that there are these little red circles, which are speckled throughout the centrosome matrix. And these are nucleating sites, or organizational sites, from which microtubules can grow. The nucleating sites are made up of a protein called gamma tubulin. And over here on the right, you can see what it looks like when microtubules are growing out from those locations. So in dark green, you see the microtubules here. And if you take a look at the orientation, notice that they've labeled the different ends of the microtubules, and notice that all of the plus ends are pointed away from the gamma tubulin rings. This means that the minus end is attached to the gamma tubulin ring. To sum that up, each of the gamma tubulin rings is a starting point or a nucleating site for the growth of one microtubule. And basically, so that word nucleating, so it's not talking about the nucleus of the cell. Nucleating just means the start or the center point. And so this is a location from which polymerization of a microtubule can start to happen. And we'll talk a little bit more about that on a different slide. But right now what I want you to know is that only the alpha portion of the alpha-beta heterodimer, the tubulin heterodimer, can bind to the gamma-tubulin ring. And basically what that means, that's what gives us this directionality. So the minus end is secured right here, and then that means polymerization can start to occur on this plus end and start to grow. Notice that the microtubules that are shown here are many different lengths, so this doesn't all happen in a coordinated fashion. Each of these microtubules acts on its own, and so some of them are going to grow and shrink, and others of them will just continue to grow. And again, from the minus end, which is embedded in this gamma-tubulin ring, then that side, the minus end, cannot grow or shrink. One fun fact for you, so the centrioles are not required for nucleation or for microtubules to grow. In fact, and this is the fun fact, I think, is that plants don't even have centrioles. They do have centrosomes, though, and they do use microtubules, but they don't have centrioles. And that is one way that we know that centrioles are not required for nucleation or polymerization of microtubules. I want to talk a little bit about why the ring structure of gamma-tubulin is so significant. Basically, you already know that microtubules are a hollow ring and that they have 13 protofilaments that all fit together in this ring and then have very long extensions going off of them. So the gamma-tubulin, again, is a ring, and so basically each of the tubulin heterodimers can start to bind to locations on that ring. So we can have these, and just pretend these are heterodimers like this, bind all the way around here, and then continue to add on top of that. So this is not a coincidence. This is one of the key reasons why these are such good microtubule organizing centers, is that it's really hard for the alpha-beta heterodimers to find each other and form a ring in just free-floating cytoplasm. So I like to think of this in terms of an analogy, like the following two things, one of which is these skydivers. I've never gone skydiving, but it looks kind of complicated to me to form this ring with all these individuals that are more or less free-floating. And I would imagine that this individual over here that's going to join the circle, it's going to be easier for that person to join than for the first couple people to find each other. But that, and I don't know if that's actually true for skydiving, but that is true for alpha-beta heterodimers to find each other and form this hollow tube-like structure. Another analogy for this is building a house of cards, and so this looks like a pretty sweet trick right here, which I have definitely never known, but the idea is that, at least for me, when I've built a house of cards, it's really hard to get the first couple ones to stand upright, but once those stand upright, then the other ones can sort of start to come together much more easily. And so that's the idea with this forming this ring as well, is if you can build off something that's already got a ring structure, then the whole process can go along much more quickly. And it looks like they even have kind of a little cheat here where they're kind of bracing things. But again, that's an idea of finding some way to simplify the process instead of having it just kind of a free-for-all. And the idea about what's going on thermodynamically is that the chance of stabilizing the structure is going to be much better when there already is a stable structure, which can form a scaffold and make it more likely that as the heterodimers each bind individually here, that they'll find each other and then form a stable structure. So one other thing I want to point out is that the microtubules can form into their hollow rod structure without gamma-tubulin. However, really this can only happen when there's a high concentration of tubulin heterodimers. And the cell typically keeps tubulin heterodimers at a pretty low concentration. One more thing, so this figure over here on the left, this is not from your textbook, but I thought it was kind of useful to check out. Notice I crossed out these accessory proteins. There's always some other accessory protein, and we're just going to ignore those. But the main thing you should see is that here we've got alpha tubulin and beta tubulin that are put together into the protofilaments. And then here we've got the gamma tubulin ring, which they're showing in blue, but that's basically what this red structure is right here. And we would be able to label this as the plus end, because that's where the beta tubulin subunits are, and the minus end down by the gamma tubulin ring. We would know that from just what I told you so far, but also from this drawing. You could pick that up. So here's a C. elegans cell. So this is a nematode, it's a model organism, and it's fluorescently labeled for 3 different cellular structures. And so my question for you is, what does each color label as far as a cellular structure? Notice that I'm asking for a cellular structure, not for a protein. Pay attention to this on quizzes and on exams. These are things that are easy to get tripped up with, but it is important to know which is which. Okay, so let's check out what we've got going on here. We've got something in blue, we've got something in green, and then we've got something in red. So pause the video and decide what those might be. Okay, so in green, that's probably the easiest one. Those are microtubules. So those are cellular structures. Each of them, you can see, very long and skinny. And then in blue, we've got kind of this shorter structure. This is actually the centriole. Each of those is a centriole. And I haven't told you yet that centrioles are actually also made up of microtubules, but they're put together in a different way. Instead of being very long and skinny, they're put together in a particular iteration here. So this, which I've just bracketed, that's one centriole, and then you can see the other centriole over here. And last, in red, this is labeling the gamma tubulin ring complex. And you can see that those are very skinny, so there's lots of them all speckled along here. And from each of those, if you can find one that's kind of isolated, like over here, you can see that there's a microtubule, just a single one growing out from it. And over here you might just see one, which has got just a real short microtubule that's present at the moment they took the picture at. Okay, one more thing, which is to say, so these are all cellular structures, so those are large, and they're made up of multiple proteins. However, if I were going to ask you about which proteins were found here, for the red, you might say gamma tubulin, that's a protein, but then the whole structure is the gamma tubulin ring complex. For microtubules, you might say alpha tubulin or beta tubulin, and we haven't talked about what proteins make up centrioles, but you would probably guess from what I've told you here that that's some sort of tubulin.